should be in. Okay. 1, 2. All right. First of all, any questions that anybody wants to start off with?
Yeah, actually, I've got a quick question.
Go ahead.
So for the second ed puzzle, there's the first question that asks you, well, it's telling you to answer cells work together to form what? And I said all of the above. It says
Should be
tissues,
tissues.
organs.
Tissue.
Huh?
Tissues is the answer.
Aren't they working together to form everything? I mean, they're forming the tissues are forming the organ systems.
Right. This is exactly what you just said. Cells form tissues, tissues form organs, organs form organ systems. That's why I think 2 weeks ago we did the hierarchy, the levels of organization. There was one day where I threw that in there.
Oh, all right, yeah. But
cells form tissues.
Aren't they all made of cells, though?
They are, but they're talking about the hierarchy. Cells collectively form tissues, and then tissues collectively form organs. Not all cells become organs, right?
Yeah, no, they need tissues first.
Well, not all cells, your blood, like for example, your red blood cells, they don't form a tissue. They just randomly float around in your blood collecting oxygen and delivering that to the other cells. But your cells like, skin cells, they collectively form, a barrier. So they form an organ, which is your skin. That's your integumentary system. So not all cells form all those things. That's why that answer would be incorrect.
Oh, okay, that's what I was missing there.
I think that's what you're looking for, because I get it. You're saying, well, don't cells make up these things? Yes, but not all cells make up. Not all cells turn into tissues, and not all cells turn into organs, and so forth.
Oh, okay, all right.
Good question. Good question. I'm hearing you. I got it. Anything else with regard to that? Mallory, just jump in if you have a question.
Okay. I don't think I have any yet, but I'll let you know.
Okay. So just and it's just easy for me, just really quick, just to recap week 7, I'll do it. And I won't even go through the stuff. I'll do it really easy for you. So week 7, the idea is we transition from cell transport to this idea of other things that would make it hard for stuff to get in and out of a cell. And that is how big a cell is. And so once we establish that the size of a cell is limited by this relationship between surface area and volume, and it's a hard concept for a lot of kids, but then once you understand that the reason that's an issue is because as the cell or any object increases in size, its volume is increasing at a rate that is higher or faster than the surface area. In other words, for every object that continues to get bigger, the space that it has to feed a cell, for example, becomes bigger and bigger compared to the surface area. So even though the overall surface area gets bigger, it's not keeping up with the pace of the volume. The only cell that does that, or I shouldn't say only cell, the only shape that did that very well was the cylinder. And that's why the last question in that activity was explain or identify which shape cell is best for muscles, and that would be a cylinder, because you're getting, in other words, in the largest cells, the cylinder had the highest surface area to volume ratio. It was like 5-1 compared to the others, which were 1.5-1, which makes sense because muscle cells need to get oxygen and carbon dioxide in and out really fast. So that's where we leave you on week 7. Now what we're doing is we're jumping into week 8. We're trying to get into the genetics. And what we're trying to do is we're trying to establish, okay, now that we recognize that cells can't grow too big, that means they need to divide. And instead of being 1 or 2 cells, we're made up of billions and trillions. Okay, so now how the heck do they do this? First of all, what is the brains that makes this process happen? 2, what is the actual step-by-step process, which is really not that important. But 3, how do they do it and not screw it up for trillions and trillions of times? So what we're first going to do, and there's a lot this week, is we're only going to touch on bits and pieces. There's no way in this setting we're going to go to that depth. The first thing we're going to have you do is to sort of understand what the brains of a cell is. And it kind of seems kind of like not the right sequence, but it'll make sense. And that is basically the structure of DNA. So the first thing that you have to do is really look at the notes and look at the structure of DNA. There's actually not a tremendous amount of notes on the structure. What's really hammered here is this thing called the cell cycle. So although I want you guys and everybody to know what DNA is made of, memorizing that stuff is not really important. What's really important is understanding the cycle of cells. In other words, when do they start to decide, maybe I should start making copies of my genetic information? And then what does that process look like? And how long is that process? And if we have time, the cool thing with it is you can get an understanding for how cancer works. Because the cell cycle literally is the issue with cancer. It's all about disruptions in the cell cycle. So what I'm going to do is I'm going to pull up the notes, but I'm going to pull them in my PDF. So I should be able to write all over this stuff. Oh, cool. Okay. So let's do this. Let's go. And I don't think I need this. Okay. Awesome. Mitosis, cytokinesis, not really where I want to start. All right, let's first figure out what this DNA thing looks like. Like I said, there's not a lot of information about DNA, so one of the activities you're going to do coming up this week, we'll hammer all that out. But when DNA gets ready to copy, it's going to look like a structure like this. It's what we call a chromosome. And your body is made up of so many pairs of chromosomes. We have, does anybody know how many pairs of chromosomes we have? No.
We have 47 chromosomes, don't we? Or 46?
46. Yeah. So we have 46 pairs of chromosomes. And this is what we would refer to as, well, this would be, we just call this one chromosome. The trick here with kids understanding chromosomes is really what's happening in a cell. So let's just do a quick circle here. And I don't know why they don't show it like this, is really you've got a chromosome there and you have, so we'll call the red one from dad, and then we'll just change the color. Actually, we'll make the red, you know what? It doesn't matter. We'll just make a blue one. And we'll go like that. And then let's just do another one. Let's do a short one here, like that, and so that you really understand what's happening here. And maybe we'll do, let's see, put it up here. Oh, sorry. All right. It's a little temperamental. I really hope. All right, perfect. That'll work. Okay. So really when you're looking in a cell, the chromosomes are based by their size. So if you were to look inside a human cell, you would see 46 pairs of chromosomes. You'd see 92 total chromosomes. If you look at the picture I just drew, you'll notice that there's this blue one here is about the same size as this red one. So 1/2 of a chromosome is coming from your mom's side and 1/2 is coming from dad's side. We can just pick whichever color. It doesn't matter. We'll say this is, I think we said this was mom, this was dad. It doesn't, but yeah, it doesn't matter. The other chromosome you'll notice is a little bit smaller. And that's exactly what happens, is that each of the chromosomes in our bodies has different amounts of genetic information on them, and they're actually different sizes. So in order for an organism to work, it needs the chromosomes from mom, but it also needs the chromosomes from dad. The picture you're looking at on the bottom here is actually what happens when a chromosome makes a copy of itself. So they show this image all the time, and kids walk away with this thinking, oh, that's the chromosome pair. That's 1/2 from mom and 1/2 from dad. No. That is not what that's showing. That's actually showing, this image is actually showing that this chromosome, let's say I had this one here. Am I in the red? I am. And this chromosome made a copy of itself It might do something like that. And then this chromosome might make a copy of itself. And then this one might make a copy of itself. So these are actually the duplicates getting ready to split apart. And that'll become clearer. I just want to point that out because you see that image so many times that it makes it confusing for students. So what they're saying here, genetic information is passed on from one generation to the next on chromosomes. It just means that our DNA is cut into segments. And that makes sense. If I was going to go to the copy room and I was going to make a copy of different activities and put them all into a packet of 46 activities, let's say, I would probably have the activities divided and I'd make copies of those before I stapled it all together instead of one giant packet of 46 because the paper jams. I mean, if you think of a copy machine, it kind of makes a little bit of sense. And before cell division, all the chromosomes are duplicated. So cells don't do this on the fly. They literally prepare for the time where they know time's up. I'm a dead cell. I better split so that whatever thing I do continues to go on, you know, maintain homeostasis with the environment. There are some features that you should be aware of. Remember I said these are copies. And so this is actually the best description. This is what we call sister chromatids. I don't know why my line here. There we go. Sister chromatids. And that's the word we refer to when a chromosome makes a duplicate copy of itself. So if you can imagine this cell again, and we've got one chromosome from mom and one chromosome from dad, then if I'm going to make a duplicate cell, I'm going to need to make a copy of that chromosome from mom. That's what I did here. And I'm going to need to make a copy of the chromosome from dad, which I haven't done because my color. So if I just go ahead and choose the color and I go like that, now I've made a copy of that. And so now the trick becomes if I can arrange these chromosomes in such a way so that this cell, when it splits in half, I now have two new chromosomes, but I got to make sure I have that original chromosome from dad and that original chromosome from mom in each of the cells. That's what the duplication is. And we refer to your chromosomes when they make a duplicate copy of themselves as sister chromatids. And they're joined together right here in a structure that's called a centromere. I know these seem strange, but the reason they're important is what eventually you'll learn. And if I could, we'll just stick with, let's go with black. What eventually you'll learn is that your body, when your cells get ready to divide, they will form additional structures, which probably aren't shown in here yet, but they form little structures at the ends. And these little structures will kind of go like this. And they're called spindle fibers. And their job is to sort of, if you can imagine, pull these things apart. And one of the things that they do in cancer cells is they can interfere with this process so that they don't get pulled apart. But what ends up happening is these spindle fibers, when the cell starts to elongate and this end pulls in this direction and this end pulls in this direction, it'll pull these two apart. And then you'll end up with two new cells, each one having the original DNA strand. And so the other one looks like an exact copy. That's what mitosis is. That's how you repair all the cells in your body. The only cells that go by a different method are the sex cells. Stop me, please. Is there any questions so far? You're just digesting. That's okay. That's fine, because that's where you should be. You should just be digesting the information right now. So all I want to make sure you know is that DNA exists in your body in structures that are coiled together called chromosomes. There's a certain number of them, and they need a dad version and a mom version to make any of the genes in your body work. Before cells divide, they duplicate that genetic information, and the duplicates have a term. They're called sisters. Sister chromatids. So instead of sister chromosomes, it's sister chromatids. And they're attached at a structure called a centromere. This sister chromatid eventually ends up split into two new cells. We don't know how yet, but that's what we know. We got a pretty vague idea of how these new cells might be similar to each other. Let's skip through this. All right. So now, and you might be like, what? That's all we want to know with DNA? Kind of, because to be honest with you, all that other structure and stuff isn't really important. I mean, it is, but not for the average person, who is out there experiencing genetic disorders and things of that sort. Knowing this process is probably the most valuable. It's called the cell cycle. And the cell cycle is basically the birth of a cell, its history of its life, the moment it says, uh-oh, I'm not doing as good as I should, so maybe I should prepare for the end of my life. And then the end of its life almost happens, and just before it ends, it duplicates itself, makes an exact copy, and the cycle continues. And it's divided into two phases. There is one phase called interphase. So these are really they're really not challenging. I just think in a classroom setting that sometimes there's a lot of distractions, and this table looks intimidating. It really isn't. So the birth of a cell, where we're going to start is right there. We're going to start right there. And I am going to just change my color so it's a little brighter. All right. And we're going in this direction. And so we've got this first phase called the G1 phase. That is the first growth phase. So remember we said cells grow to a certain size? In that phase, that's what they're doing. Little baby cells, and they're developing and they're growing, and they're almost generally the size they should be, but they're still establishing stuff. And so there's organelles and features inside a cell, and every cell is slightly different, complicated, but in every cell they're growing. And in that beginning process, they grow, which does not take very long. And then they begin to do what they do. If you're a pancreas cell, at the right time, you're producing insulin. When you eat, you need insulin to open up the feedback mechanism. If you remember, there was facilitated diffusion. Insulin goes in, opens the gated channel, glucose goes in, and then once you're full, the brain sends a signal, tells the pancreas to stop doing that. Okay, so let's talk about a pancreas cell. It's producing insulin, and it's doing a great job. Now, every cell has a certain life to it. Blood cells live for 90 days. All cells live different. Like some of them never die, like your brain cells. They only die when you die. So whatever damage you do to them, they don't reproduce. That's the key there. So we've got this cell, and it's doing what it's doing, and then all of a sudden, its efficiency starts to deteriorate. Could be getting too big. Okay, could be just old, wearing down. Could be a trauma, maybe an accident or a toxicity of something that causes trauma. Repeated abuse of alcohol and drugs, they do cause trauma to your body. And that signals those cells to say, uh-oh, I'm going to need to reproduce because my body is saying you're inefficient, and I'm going to take these other cells and eat you up because you're garbage, and we're going to get rid of you. But before you do that, I need to make sure your job is filled by a new cell. And so the cell, now look how much time, all right? This is all parts of time here. So it spends this time in this S phase. It starts to replicate. And during the S phase is when this happens. This is during the S phase. Not all this other junk I put in there, but the actual chromosomes, the duplication of your DNA, the coiling into the chromosomes. Now, some of the stuff I'm giving you here is, if someone were a bio teacher, they might actually point out some errors. Like, for example, the actual coiling of DNA into a chromosome really happens here, but I tell kids it happens here just because they need the visual, because it's hard to imagine your DNA inside your nucleus like spaghetti. I mean, like really, if you think about your DNA, if this is your cell, your DNA is all like this, and it's duplicating. Like that's, almost impossible for someone to observe. How does the thing of spaghetti duplicate itself? So the S phase is this duplication. So if you were to look at a cell, which unfortunately we don't do, I don't know if I'll incorporate it, but if you were to look at the cell under a microscope, you would see, let me see if I can just quickly pull this up. Yeah, because it's worth it. It's totally worth it for you guys to see this. Let's see. I'm going to go with here, lab activities. We're looking for root lab. That is it. And let's view extra large icons. And I think interface. So, oops. All right. And I always don't really know if I'm in interface, but what you see is the cell in its little life cycle. In this process, it's starting to duplicate DNA. Whoa. That was cool. It was a hawk that just went right by the window. That was really cool. You'll see their cells are all in different phases in here. Interphase, this is the nucleus. As the cell goes through interphase and it goes through the synthesis phase, you would see the nucleus more condensed. Let me hide this out of the way. Let me back out. Let me just, should be another one here. I thought there was. There we go. Yeah, that's what I'm looking for. So you can see here, and then you can see a little bit of like coiled pieces. That's when you know like the cell is starting to go into the prophase. The nucleus is gone. All the DNA is in the space there. So, and you can actually see like the DNA separating. These are different stages of mitosis. And then when it separates into two new cells, these are plant cells, by the way. And you can see they have a structure to them. Human cells, animal cells wouldn't look like this. The reason we always use plant cells is because they have a cell wall. So the cells maintain a nice rigid shape. But visually, if what we're talking about through a microscope would look like this, if we looked at all the stages. And interphase is right in here. This is before anything divides. Okay, so now the cell does that. And then you'll notice there's a second growth phase. And so that's when the cell says, all right, I am ready. I'm ready to divide into two cells, but I'm going to continue to do my job until that time comes. And so it makes sense that it's a little shorter than the initial growth phase. And then it happens. It's a little bit bigger. Once you go through your life, which is interphase, then you go through cell division, which is called mitosis. And mitosis has an order. They sometimes refer to it as PMAT. So PMAT, and then if you want, you can put cytokinesis. And so PMAT, prophase. And so I'll just sort of, before I show you all the steps, actually, we're not going to do all the steps yet, but just let me like kind of walk you through a visual, like a storyline of it. Prophase would be those chromosomes that I showed you that are like all coiled. So you've got your chromosomes, they're like this. Sometimes they just do X's. And then metaphase is when the chromosomes line up in such a way that if they were to be split, it would create two new cells with the identical information. So remember we said our cell has, let's change the color. We'll just do two chromosomes. So our cell has two chromosomes, and then it has the copy. All right. So how could I arrange this so that each cell gets exactly the same information as the original cell, which was one of these and one of these? And so the way your cell does that is it takes this arrangement down here and it makes one like that. And then it takes the other one and puts it underneath it. And all this is happening inside the cell. And then remember those spindle fibers I told you about? They're the ones moving all this stuff around. So when I'm referring to spindle fibers, what I'm referring to is this stuff. This stuff here is moving all of these chromosomes all over the place. They're actually manipulating this pattern. So once your cells line up, if your cell was to split in half right here, and this half went this way, and this half went this way, you would end up with 2 new cells. One would have this chromosome half, and one would have this one. So you'd end up with a cell that had this and this, which is exactly what's up there. And then the other side would have one of these and one of those. And so if this were a skin cell and you had gotten a cut and you needed to reproduce the skin cells, this is what's happening. You have 2 new cells compared to your original. And it does that with all pairs of the chromosomes in your body. Now, what's really cool is this arrangement I'm talking about. Think of, now this is mitosis, but when you get into meiosis, the arrangement, this piece here, how they're arranged, changes, and you can change which ones are on which side. So all of a sudden you have all this line of all these chromosome pairs, and you can switch them. So imagine being able to take the mom side and the dad side. Nothing says that the mom side has to line up on the left and the dad side on the right. You know, your chromosomes are moving around. You might have one chromosome, chromosome number 1, that has mom's chromosome on one side and dad on the left. And then chromosome 2 has them flipped. But you can flip number 3, maybe you can 3 through 30. I mean, you can flip them all. So that creates all the variation. There's other cool stuff that your body does too throughout the process, but I don't want to lose you in it. This video is a resource. I think it's in today's. If it's not, it's definitely in tomorrow's. It's a video source you're definitely going to want to watch. I probably put it in for tomorrow because I thought there were too many videos that you needed to see. So here's the explanation that I gave you so that you have something a little bit more refined. G1, that's your first gap. S phase, that's your synthesis phase. G2, and then mitosis. And these 3 phases are what we call interphase, and that is basically when the cell is living. And then mitosis is the process of cell division. This gives you a little bit more information. Basically what I verbally said about the growth, the synthesis, the chromosomes, duplication, the second half of growth phase, and then you get into the 4 stages of mitosis: prophase, metaphase, anaphase, telophase, and then cytokinesis. And I believe we pretty much stop there because this is going to be at the end of the week. Worth noting, just if you're interested, remember I said that your DNA in your cells are wrapped up like spaghetti. Your cells actually manufacture a protein, and that protein allows the spaghetti, this is your DNA spaghetti-like, actually allows it to coil up on these proteins. So if you can imagine all these proteins being all bunched together, the DNA coils on that, and that coiling is what actually creates the chromosome. So here again, just as a repetition, this chromosome, say, comes from dad, and this one comes from, oops, whoa, I don't want to do that. This one comes from dad. This is the duplicate. This is what we call sister chromatids. Right here would be the centromere, and these are sister chromatids. They are a duplicate of each other. That's why I put dad for both. All right. Questions, please, anything. Something that is not clear. All right, let me go back then to today's assignment just so that we know, like, okay, so how does that tie into what we're doing with today? I had you look at the structure of DNA, and I had you do that in an ed puzzle. Very little of what I talked about today has to do with the structure of DNA. And most of that reasoning is because I don't think it's super important for you to leave here with that. Oh, don't get me wrong, it's important because there are other for understanding cancer and mutations, understanding the genetics of how DNA, genetic is redundant, understanding how DNA actually makes a copy of itself. Like at the chemical level, how does it actually make a copy? And it has a unique way of, first of all, its shape has oh, this isn't my pad. Sorry. Let's go take a look, actually. Make it easier rather than try to draw it. All right. So your DNA actually has this double helix shape to it. And what's really, really cool is the DNA actually splits in the middle here. And so this whole thing just splits in half all the way through. And it goes in one direction. So you have two splits. So now I've got a ladder that looks like this that has these runs that are coming off of it. All right. And we've got this other side that has these runs that are coming. Oh, 1, 2. I didn't line them up right. Darn it. 3, 4. There we go. So we've got these runs that are coming up. And so what ends up happening is this side actually makes an exact copy of it, this side. And this side makes an exact copy of that side. So you end up with two new ladders over here, 1, 2, 3, 4, and on that side that are actual copies of one another. And the cool thing is it does it with only 4 letters, 4 things called nucleotides. They happen, I'm just going to put it in here, like I said, just for the heck of it, because you're here. There's one that begins with the letter A that only forms with T. And then there's another one that begins with the letter G. Oh, my arrows are not working because I have this thing on. Can I take that off? It's not. Whatever. And it only forms with letter C. So if you were to make, and I'm going to run out of space here, but let's see. Let's say this run had an A. It's hard to see. This side here would be T. And let's say this had another A. This side would be T. And let's say this was a C. That would be G. And if this were a G, over here would be C. So now what happens is this side of the ladder will make a duplicate of itself because T's only form with A's. So chemicals come in here and they basically make a new strand on this end that'll be A, C, G, which now will duplicate the whole thing. And this strand will make this side T, T, G, C. And you end up with 2 complete copies. And it does some other cool stuff, like one direction, like this side may form from this direction, and this side may form starting from the bottom up, while this one goes from the top down. That's called anti-parallel. That's so cool because if you think about it, imagine you and I both started at the same direction and we had to assemble. So now imagine a bunch of cones, and there's all these cones, and I have to put the correct letter cone next to the cone that I run by. And you're putting the A's, and right, you're on one side and you're running down going A, C, G, and I'm right next to you and I'm putting T, T, G, C. If there's not a lot of space here, we're bumping into one another. Well, wouldn't it make so much more sense if I said, hey, why don't you start way down at that end of the DNA, and I'll start way down at this end of the DNA, and I have all this space, and I can go ahead and boom, 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 boom. Well, you're going this direction, and the only time it becomes a problem is when we meet in the middle. And of all the strands of DNA, if there's that one time when we meet in the middle that's a problem, that's not too bad, because the alternative would have been we would have been in each other's way the entire time. And DNA is really, really long. So that's way more information than you need. But as you go through the activities, like the Ed puzzle today, this is the kind of stuff that will, like, it'll click. You'll be like, oh, okay, I get it. I see where he's going with this. And the Amoeba Sisters does a really good job of like the pacing in there. All right, questions? Crickets. It's always crickets.
I think I'm good.
All right, so, go ahead. Did I do good?
Yeah, you did. And I watched. I did the first Ed puzzle already, so this makes it. It makes more sense now. What you just went.
It does, right?
Yeah.
You got to do the live sessions. If you don't do the live sessions in this unit, you're dead in the water. I don't see how any kid could teach themselves this stuff. Without the stuff that I just did and try to like give you those analogies, as poor and as silly as they are, because sometimes I don't use good ones, I just don't see how a kid could really absorb that and even be able to come up with those analogies. Before you go, great question, by the way. Thanks for the compliment, Mallory. Tomorrow, it still says structure and replication. This is a POGIL activity. Now, it's no surprise that these things aren't on the internet. Everybody knows that. This is a challenge one, but I just want to point it out because, boy, I wish I could give you a jump on this, but it'll be tomorrow. You can see these are the letters I was just referring to. I don't have it open on my thing, so I can't use my highlighter, but these are the letters A, T, G, and C. And what they did here is instead of just giving an arbitrary A's go to T's and C's to G's, they did it in a way that makes sense, that is in sync with what we did in the feedback loops and stuff, or the facilitated diffusion especially. See the shapes? You can see that A's only connect to T's because of the point here. And you can see that the guanine has the curved shape and the cytosine has the curved end. So you can see how guanine and cytosine, C's and G's, only go together. And so the shape is what determines which one. That's why A's and T's only fit. The other ones don't go in. So now as a DNA, I can ensure that if I make a copy of myself, that it'll be an exact copy of it because there's only so many letters. There's only one letter that'll fit. Now, what's really neat is you're telling me that all of the DNA, all of the features that make you, me different from each other is based on these combinations of 4 letters? Absolutely. That's exactly what I'm telling you. What's really cool, though, is that there are sequences. So your genes are sequences of these letters. Complicated genes are longer sequences. Less complicated genes are shorter sequences. So if you were to get into higher end biology, you would learn what's called a codon. It basically tells in your strand of DNA. So, you've got this, oh, I can't draw on this, but you got this super long strand of DNA and on it may have 5,000 genes. Well, there has to be some way for your body to know when the genes are, like what portion of the DNA is which gene. There's got to be a start and stop for each gene. I get it. It's 1,000 genes long, but where does gene 1 start and stop? And your DNA has signals. They're called stopping and it's the AP bio stuff, but there's stop signals and start signals. And that's basically how your genes are set up. Where mutations come in is if something goes wrong in this sequence and you have a gene, a trait that has a combination of these letters that may be 600 or 700 of them long, and one of them gets flipped or one of them gets flipped front and back, then that gene may not function correctly. And that's what a mutation is. And it happens. Your body does an awesome job of eliminating that possibility, but it happens. But that's why mutations are so few and far between. Generally, if the mutation isn't viable, meaning it doesn't allow the organism to live or reproduce, then you won't see it in the population. You see it in humans because we take care of our sick. So as you go through that tomorrow, there's that anti-parallel. DNA gets copied in one direction on one side and one direction on the other side. Hydrogen bonds, a term from Unit 1 and 2, that is just the force of attraction between the substances. They're not actually molecularly combined. It's just the force of attraction. It's like what keeps water combined with other water molecules. This is a little bit more complicated process of how DNA splits and chemicals come in and assemble along the 2 separate strands. And then, this is not really that important. There's the cell cycle again with just a little bit more detail. And I think it pretty much ends with a couple of, that's it. It ends right there at number 20. Pay attention to number 20. This question is on a unit check-in. So this is really important because kids need to know what they're looking at when they see how chromosomes are written when they're copied. That's pretty much it. I will take this right now, and this session, I'll put it right into WeVideo, and I'll get it uploaded as soon as possible because I have a feeling this is the kind of thing you want to have on your computer to pause and stuff to be able to refer to. So I'm assuming everyone has other classes to get to. So if you don't have any additional questions, certainly can sign off. If you have other questions later on today, send me an email. I'll be around, and this will be within the next hour and a half. I'll have this posted on the live stream for you.
Thank you.
Excellent. Okay, we'll stop presenting. And we'll stop recording.